This is the fourth video in a series about the Golden Gate cloning DNA assembly method. In previous videos, we learned how type 2S restriction enzymes work, how to use them in one-pot, one-step Golden Gate cloning reactions, and we also learned about the modular cloning or MoClo Golden Gate assembly standard. In this video, we are going to learn how to make new level 0 parts that are compatible with that assembly standard. First, let's look at a level 0 plasmid and see how it works. This is a level 0 plasmid acceptor for promoter parts. According to the Golden Gate MoClo assembly standard, which was fully described in the third video in this series, promoter modules begin GGAG and end TACT. The level 0 acceptor plasmid contains a LAC-Z cassette flanked by inverted BPI1 recognition sequences. When this plasmid is digested with BPI1, the LAC-Z insert will drop out, leaving 4 base pair overhangs. To clone a new promoter part, illustrated in green, into this gap, it needs to have compatible overhangs. To do this, we amplify our desired sequence using primers with 5' overhangs that introduce two things. Firstly, BPI1 recognition sites, and secondly, 4 base pair sequences to make our compatible overhangs. BPI1 requires 2 base pairs between the recognition site and cut site. These can be of any identity. The PCR amplicon flanked with the required sequences and the level 0 acceptor can be mixed together in the same tube and a Golden Gate digestion ligation or Diglig reaction carried out. In this reaction, the BPI1 will remove both the LAC-Z cassette and the ends of the amplicon, producing compatible overhangs that the T4 ligase will stick together, producing our level 0 part. If you need to review the Golden Gate one-pot, one-step Diglig reaction, please watch the second video in this series. The new level 0 part does not contain any part of the BPI1 recognition sequences. The part is now flanked by 2 inverted BSA1 recognition sequences, features of all level 0 acceptor backbones. Digestion with BSA1 will release the part with the same overhangs, ready to assemble with other level 0 parts. All level 0 parts can be cloned in the same way, as there is a corresponding level 0 acceptor for each type of standard part defined in the MoClo assembly standard. So now, let's take a look at how to make new standard parts. There are 2 things to consider: primer design and the removal of internal BPI1 and BSA1 recognition sequences. Primers for each type of module will have the same structure. Each will consist of a random base pair so that the BPI1 site is not right at the end of the amplicon. In this example we have used a T. The BPI1 recognition site GAA, GAC, 2 base pairs to bridge between the recognition site and the cut site, the 4 base pair overhang, which is defined by the type of standard part being made, 18 to 30 base pairs that will anneal to the sequence being amplified. The length will depend on the specific sequence. A longer primer may be more specific, but it can be made shorter to avoid including a homonucleotide tract or palindrome. For non-coding modules, the design is straightforward. Promoter modules, or pro modules, can be made by including GGAG in the forward primer and AGTA, which is the reverse complement of TACT, in the reverse primer. 5' untranslated region modules, known as 5U modules, can be made by including TACT in the forward primer and CATT, which is the reverse complement of AATG, in the reverse primer. 3U plus TER modules, that is, modules that contain both a 3' untranslated region and a terminator, can be made by including GCTT in the forward primer and AGCG, which is the reverse complement of CGCT, in the reverse primer. For modules that contain coding elements, the frame of the part must also be taken into consideration. Coding sequence modules that contain stop codons can be made by including AATG in the forward primer and AAGC, which is the reverse complement of GCTT, in the reverse primer. The ATG found in positions 2, 3 and 4 of the AATG overhang in the forward primer will encode the methionine start codon. Therefore, the first base of the part should be the first position of the codon directly after the start codon. Likewise, the last 3 positions of the part should encode the stop codon. The reverse complement of the stop codon will therefore be the first 3 positions of the gene-specific part of the reverse primer. To make a coding sequence module that can be joined to a C-terminal tag, a CDS-NS module is made. The NS means no stop, as the stop codon is not included. The overhang at the end of the part changes to TTCG. The reverse primer changes to CGAA, which is the reverse complement of TTCG. The last 3 positions of this TTCG overhang encode serine. This puts the first T into the third coding position. To keep this coding sequence in frame with the C-terminal tag, the part could end on the second position of the last codon before the stop codon. However, if replacing the native position 3 base with a T makes an undesirable amino acid, then 2 additional G bases can be inserted. This will mean that a glycine and serine are inserted between the coding sequence module and the C-terminal tag module. 5UF modules contain 5' untranslated regions, but fuse to N-terminal tag or NT modules rather than to coding sequence modules. Like 5U modules, 5UF modules can be made by including TACT in the forward primer, but use ATGG, which is the reverse complement of CCAT, in the reverse primer. The last 2 bases of the CCAT overhang contribute the first 2 bases to the ATG start codon. N-terminal tag or NT1 modules, therefore, are made by including CCAT in the forward primer. As the A and T for the ATG are encoded in the overhang, the region of similarity with the part must begin with G and continue with the first base following the ATG. CATT, which is the reverse complement of AATG, is included in the reverse primer. Finally, C-terminal tag modules can be made by including TTCG in the forward primer and AAGC, which is the reverse complement of GCTT, in the reverse primer. The last 3 bases of the TTCG overhang encode a serine. Therefore, the part should continue in this frame starting at position 1. This part should contain a stop codon to close the open reading frame. As well as being flanked with the correct 4 base pair sequences, parts must also be free of internal BPI1 and BSA1 recognition sequences. If there are multiple sites, it might be faster and cheaper to order a synthetic version from a commercial gene synthesis company. If only a few sites are present, they can be easily removed, simultaneous with cloning into the level 0 acceptor. Take, for example, the coding sequence shown here. In the centre is a BSA1 recognition sequence, GGT-CTC. We have already learned how to design primers to make a CDS1 module. Now, let's look at how to make 2 further primers that will introduce a synonymous change to eliminate the BSA1 recognition sequence. First, we will decide which base to mutate. To make the mutation silent, we'd like to mutate a base in the third coding position. We have 2 choices, either the first G or the first C. In this example, we are going to choose to mutate the C. GTC codes for valine. Valine is also coded for by GTG, GTT and GTA. In theory, we could choose any of these alternatives, but there are 2 factors that will influence our decision. The first is the codon usage of the organism in which we are going to express this sequence. If we check the codon usage table for that organism, we see that GUA is only rarely used. The tRNA for GUA, therefore, might be rare in the cell and might limit translation, so we would avoid this. GUU, however, is most used. A C to T mutation might be the best choice. The mutation is going to be introduced in a new 4 base pair overhang. So the next thing to choose is an overhang that is compatible with the 2 that we are already using. All overhangs in one reaction must be different in at least 2 of the 4 positions to prevent misannealing. We already have AATG and GCTT. The position of our C to T mutation in the 4 base pair overhang doesn't matter, so we have 4 choices: TTTC, TTCT, GTTT and GGTT. All of these choices are different in at least 2 positions from both of the overhangs already in use. Let's choose TTCT as it is only similar to one overhang at one position. Next, we are going to design 2 primers to use with the forward and reverse primer already marked. These will allow us to amplify the coding sequence in 2 fragments. The first primer will be used with the existing forward primer to amplify the first half of the sequence. The principle is exactly the same: a BPI1 recognition sequence, a 4 base pair overhang, this time the custom overhang to introduce the SNP, and 18 to 30 base pairs of the gene-specific sequence to allow the primer to anneal. The second primer will be used with the reverse primer to amplify the second half of the sequence. The next step is to perform 2 PCR reactions with our primers. This will provide us with 2 amplicons. These 2 amplicons can be cloned into the level 0 acceptor for CDS1 modules in a 1-pot, 1-step Golden Gate cloning reaction. BPI1 will cut both amplicons and the level 0 acceptor, leaving compatible overhangs. The T4 ligase will ligate the overhangs together, making our final CDS1 part free from internal BSA1 recognition sequences. That's the end of this video. If you missed information on how type 2 restriction enzymes allow 1-pot, 1-step assembly of Golden Gate parts, or need to learn more about the Golden Gate Moclo assembly standard, please watch the first 3 videos in this series.